Hello, my name is Meyers, and I'm an undergraduate biomedical engineering major at Michigan Tech. During the summer of 2019, I received a fellowship from the Focused Ultrasound Foundation Global Internship Program to work on an ongoing collaboration project between a biomaterials lab group at Michigan Tech and a focused ultrasound lab group at Virginia Tech. Our project consists of using focused ultrasound to remotely modulate nitric oxide release from an injectable hydrogel for tendon repair. First, here's a little background on our project. So, tendon injuries and tendinopathy affect normal tendon structure and function through the degradation of tendon tissue extracellular matrix, or ECM. Currently, tendon injuries are conventionally treated with reconstructive surgeries, physical therapy, and steroids. However, there have been some recent developments in the field, including growth factor injections and platelet-rich plasma treatment. These treatments still have many drawbacks, though, including long recovery periods, the formation of adhesive scar tissue, and susceptibility to repeat injury. As a complementary or alternative approach to treating chronic tendon injuries, our group has been working on developing injectable adhesive nitric oxide releasing hydrogels to help restore tendon tissue structure and function. Looking at panel A, these composite hydrogels are synthesized with fibrinogen, a protein involved in natural wound repair during homeostasis, functionalized polyethylene glycol, or PEG, a biocompatible synthetic polymer commonly used in tissue engineering applications, and SNAP fiber microparticles, where SNAP is our nitric oxide donor. Nitric oxide, or NO, is a naturally antimicrobial signaling molecule with a short half-life. Since SNAP, our NO donor, can controllably be cleaved to produce NO in response to thermal and mechanical stimulus, we aim to use focused ultrasound, which provides the perfect combination of these two stimuli, to control NO release remotely from our hydrogels. In panel B here, you can see our approach of using focused ultrasound to control the concentration of NO and fibrin degradation products released from the microparticle component of our hydrogels. Both NO and fibrin degradation product concentration are involved in the regulation of ECM synthesis and degradation. For example, panel C shows that relatively low concentrations of NO and fibrin degradation products activate matrix-degrading enzymes called matrix metallothionein proteases, or MMPs, whereas relatively high concentrations of NO and natural tissue inhibitors of MMPs, or TIMPs, inhibit MMP activation and therefore ECM degradation. To test the ability of NO release from microparticles to influence MMP activity in tenocytes or tendon cells, initial studies with constant thermal stimulus provided by an incubator were performed. The graph on the left shows the real-time release of NO from varying dosages of our microparticles incubated at 37 degrees Celsius in cell culture media. On the right, gelatin zymography results show that relatively lower dosages of NO-releasing microparticles encourages MMP activation, whereas higher dosages of NO-releasing microparticles inhibits MMP activation. The concentration-dependent effects of NO on MMP activity in this study are also consistent with the literature. After verifying that NO release from our materials had the potential to modulate MMP activity at constant temperatures, our goals were to develop a focused ultrasound system to thermally and mechanically control NO release from our microparticles remotely, characterize the release of NO from our hydrogels using different focused ultrasound parameters, and to form a basis for future in vitro and in vivo models. Here's an overview of the focused ultrasound system we utilize at Virginia Tech for our studies using a 3D positioning system, 3D printed acoustically transparent sample holders, and a 700 kHz histosonics transducer. With this setup, we inserted thermocouples into different wells of our sample holders and targeted this well by warming it for 3 minutes to a temperature of about 41 degrees Celsius and maintaining that temperature for 5 minutes, whereafter the targeted sample was allowed to cool down. Thermocouple probe results indicated tight spatial and temporal control of focused ultrasound treatment as indicated by the near instantaneous transition among warming, treatment, and cooling phases, as well as minimal heating of the surrounding wells in the sample holders. At the moment, we are working on developing a focused ultrasound system at Michigan Tech using a surgical high-intensity focused ultrasound transducer so we can eventually perform in vitro and in vivo studies. We are also designing acoustically transparent sample holders that are compatible with our NOA analyzers so that we can obtain real-time focused ultrasound-induced NO release from our hydrogels. Overall, we aim to use this minimally invasive approach to controlling ECM degradation and synthesis remotely through combining techniques from both the fields of biomaterials engineering and focused ultrasound. With that, I would like to acknowledge my advisors and all of our collaborators for their help on this project, and thank the Focused Ultrasound Foundation for providing funding for this work, as well as the NIH.